Good morning, friends. So, in this lecture, I am going to discuss you about the linkage. So, we know there are two terms which are associated with each other, that is linkage and crossing over. We have uh, discussed some brief idea about the linkage when I was introducing the topic to you in my very first lecture. So today we will see in detail what linkage is and how it is affecting the inheritance of allele or gene from one generation to next generation. So let us start with linkage. So we know we have already discussed that organism has many genes. Any individual is or any species they are having many genes, and that genes are at least they are more than the number of chromosomes that animal or individual is possessing. So it indicates that in one chromosome there may be more than one gene. That means each gene is not having their own chromosome. So what will happen? It also indicates that genes may be linearly arranged on the chromosome because there are not as many chromosomes as that of the genes. So few genes they will be lined up in a row on each chromosome, and some of them are going to be located very close together. So either if they are linearly arranged on the chromosome, so either they are very close to each other or they may be very distant to each other. So as I have already discussed you about the linkage or linked gene, the genes which are very closely associated, many a times they are known as linked together, they are linked gene. So these genes, those genes which are linked together or which are very closely placed, these genes will not be assorting independently at the time of meiosis. As we know the Mendel's law of independent assortment. So what it wanted to tell that if genes are very closely associated, they are very closely located on the chromosomes, there is chance that they may not assort independently. So Mendel's second principle, the law of independent assortment, will no longer be universal. That will always not be true, as we have already discussed when I was discussing you about epistasis. So here you in this diagram you can see that in first case, this is first case and this is second case. So here in first case you can see the genes A and B. A and B are two genes. They are located on different chromosomes. So what gametes they will produce? So they will produce capital A, capital B together. They independently assort. Small a, small b together, independent assortment. These two are of parental type. Another is small a, capital B and capital A, small b. So all four combination, they will be producing in the equal proportion, that is 25% of each. Consider the second situation where genes are present on the same chromosome. They are not on different chromosomes, they are present on the same chromosome, but they are far apart. If they are far apart, there will not be any problem in combining all these four alleles together in four combination, that is capital A, capital B, capital A, small b, small e, capital B, and capital A, small b. All these four gametes in the equal proportion, that is 25% each. Now, in another situation, if you see here, the situation third. So in third situation, if you see, the genes are present on the same chromosome, capital A and A and B genes are present on the same chromosome, but they are closely associated. They are located very close to each other. So in that case, if you see the formation of gamete, the gametes are of four type. but the frequency of formation of new combination means the combined format these two capital A capital B and capital A small a and small b, these two are the parental combination, and these are the new combination or recombinant type. So if you see, the recombinant type is having frequency of only 4%, that is 2% and 2%, whereas the parental type is having the frequency of 96%. That means 48% and 48%. So this is the third condition where alleles are closely located and they will not form gamete independently, or they will not form gametes in the equal proportion. That means these two genes are not assorting independently. If they are not assorting independently, that means they are linked. So Sturtevant was the first who showed that genes are arranged in a linear fashion on the chromosome. And he observed that the two pair of allele in sweet peas, that is Lathyrus odoratus, they do not assort independently. And this was given by Batson and Punnett. When Batson and Punnett were working with Lathyrus odoratus, that is sweet pea, they observed that the two pair of alleles are not assorting independently, and they gave the concept of, although they, at that time they gave the concept of coupling and repulsion. So linkage, instead of linkage, they told it as coupling and repulsion, or they try to explain linkage in terms of coupling and repulsion. So now we'll see what is actually coupling and repulsion. So here you can see the idea of coupling. What is coupling? So actually Batson and Punnett. So Batson and Punnett, they gave the idea of coupling and repulsion, and in coupling they wanted to tell that the parental combination of gene try to inherit it together. That means two dominant allele from two different genes try to come together in the process of gamete formation. So either dominant or recessive, you can say it in either way. If two dominant are coming together, that two recessive will go together. So for that situation, he termed that situation as coupling. Thus gamete containing the dominant alleles, that is capital A and capital B, and its counterpart gamete, that is small a small b, deviate from independent assortment. So they will not independently, because if you are talking about independent assortment, they have to assort independently. They don't have any association. But Batson and Punnett told that they try to inherit together. That is called as coupling. The coupling could be partial or complete. So in the later part of this discussion, we will see what is partial or complete linkage or coupling. In 1911, Batson and Punnett discovered gamete with one dominant and one recessive allele. So again they worked and they found that there are gametes, that means new combination, not of parental type but no new combination, where dominant and recessive alleles will come together, and they term it as a repulsion gamete. So they gave the idea of coupling and repulsion, and they try to tell that dominant alleles are coming together, that is called as coupling, and if the dominant and recessive both are trying to come together, that is called as repulsion. Now we'll again see what this coupling and repulsion imply. So it is of opinion, or it is opined that coupling and repulsion is having no meaning if there is complete dominance. Complete dominance, or you can say it is a complete linkage. Coupling and repulsion is having no meaning if there is complete linkage. Or you can say that if there is no linkage, there is full crossing over, or chromosomes are located on different, genes are located on different chromosomes. What coupling is? In coupling, dominant alleles are on the same homologous chromosome, and both recessive alleles are on the other homologous chromosome. So we have already discussed that is capital A, capital B, small b will be on the same chromosome, and this situation is called as cis. So many a times you will get a question in your examination, what identify from the following that which one is the cis condition or in the coupling phase. Similarly, repulsion. So each homologous chromosome has one dominant and one recessive allele from the two genes. Thus, parental genes, but new combination, that is capital A small b and small a capital B, they call it this situation as trans situation. This is not parental gametes, but you can say it as new combination. So this is about the coupling and repulsion. I hope that coupling and repulsion is clear to you. The term was given by Batson and Punnett, and they defined that coupling means both the dominants or both the recessive alleles are trying to come together, and reverse is true for repulsion. Now what is linkage group? So based on the study, the T.H. Morgan further advances his study in the Drosophila, and in 1911 he actually proposed the concept of linkage. So actual concept of linkage, it was proposed by T.H. Morgan, and based on this linkage he gave the idea of linkage group. So what is linkage group? Linkage group means the number of haploid chromosomes present in that species or that individual. So that is linkage group. If somebody is asking what is the linkage group in camel, you have to tell that the linkage group in camel is 37. In case of suppose in case of cattle it will be 30. In case of rabbit it will be 22. Similarly, if you know the chromosome number of an individual, you can say how many linkage group that individual is having or that species is having. So now all the genes arranged on a chromosome in a linear fashion are said to be linked to one another and belong to same linkage group. So that is why the concept of linkage group come, because the genes arranged on the chromosome in linear fashion and they are said to be linked. So all the chromosome present on a particular all the genes present on a particular chromosome, they are said to be linked, and that chromosome will be called as linkage group. The phenomena of inheritance of linked genes in the same linkage group is called as linkage, and that will determine the strength of linkage. What will determine the strength of linkage? The strength of linkage will be determined by the distance between the two genes on a particular chromosome. If the distance between the two genes are higher, there will be less chance of linkage. If there is distance is very small, there will be high chance of linkage. So now, if you see here, in case of Drosophila, the T.H. Morgan observed incomplete linkage. Why incomplete linkage? We can see here, he worked on the wing color, wing shape and color in Drosophila, and he crossed two Drosophila. One is having grey and long wing, another is having black and vestigial wing. So what he told that grey and long wing is having composition of capital B and capital V, whereas black and vestigial is having combination of small b and small v. Now in the F1 generation, he observed that all the individuals obtained were having wing grey and long, but the composition or that genotype, he observed it was capital B small b and capital V small v. So, in order to determine whether there is linkage or not, we know that we have to do test cross. Why we want to do test cross? Because in test cross, we will be able to know that how many new combination or new genotype will be formed, or how much proportion of new gametes will be formed during meiosis. So here you can see when grey long F1 generation was crossed with black vestigial, that is recessive parent. When F1 generation was crossed with recessive parent, so here you can see that there will be formation of parental type gamete or new gamete. And if you see, if you combine this together, the individual should form in the ratio of 1:1:1:1, that is 25% of each. But in reality, what Morgan observed, that the new combination was 18%, whereas old combination was 82%, whereas it should have been in the ratio of 50-50 or 1:1. But he observed that the non-crossover or parental type were more in frequency in comparison to new combination. So he thought that there is case of incomplete linkage. There is no complete linkage. In case of complete linkage, all the progeny would have been in the form of parental type. No new individuals would have formed. But he observed new combination of new genotype, but in lesser frequency. That is why he called it as a incomplete linkage. Now, based on this, you may now think that there are two types of linkage. On the basis of what? On the basis of absence and presence of non-parental combination. So, in complete linkage, two or multiple traits, characteristics or traits are inherited and normally surface in two or further generation in their parental or original combination. So, in case of complete linkage, there will not be any new combination. So, what will happen? Two or multiple traits will be inherited and in the next generation in parental or original combination. Then the second is the incomplete linkage. So incomplete linkage, it will be displayed by genes that generate some portion of non-parental combination. That means in this case, new combination will be found. It is reported in the female Drosophila and various other organisms. In the last slide, we have seen the example in Drosophila, the example of incomplete linkage. Now, what is chromosome theory of linkage? So Morgan and Castle, based on their work on Drosophila regarding the linkage, they gave the chromosome theory of linkage. According to that theory, genes are located on the same chromosome. Genes located on the same chromosomes are said to be linked. Why? Because they will be inherited together. Then the linked genes are arranged in linear fashion. We all know that the linked gene, they will be on the linear fashion on the same chromosome. Then degree of linkage is determined by the distance between the genes. So what he observed, he gave in the form of theory. Coupling and repulsion are two aspects of linkage. So we have already seen the coupling and repulsion, what coupling and repulsion tell. Now, what are the factors affecting linkage? Actually, it should be strength of linkage. What are the factors that will affect the strength of linkage? So first is distance. Already we have seen that genes, those are located widely. That means they are wide apart. In that case, there will be weak linkage. With age, the strength of linkage will be decreasing. Then temperature. Increased temperature will decrease linkage. So as you increase the temperature, there will be lesser strength of linkage. Then radioactive rays, these will also reduce the strength of linkage. So these are the factors that will affect the linkage. So with this, I have covered most of the part of linkage that is sufficient for the UG students. You go and study some standard textbook, and if you are finding any problem with this, you may contact me. Thank you very much.